Hello, I am Professor Sims, and this video is going to be the introduction to my pathogenic microbiology course. There are, not including this intro, 10 lessons for this course. If you're currently enrolled, please consult the syllabus and Moodle site for assignments and other course information. First, we want to talk about signs and symptoms, communicable diseases versus non-communicable disease, different types of disease, specifically iatrogenic, nosocomial, acute, chronic disease, the periods and stages of an acute infectious disease, different aspects of virulence, pathogenicity, pathogenesis, comparing different types of virulence factors, specifically exotoxins, capsules, epidemiology and its relationship to public health, different modes of transmission, contact, vector, vehicle modes of transmission, and then emerging and re-emerging infectious diseases. This is a challenging field of science, and therefore it is a challenging course. You're going to be expected to apply your knowledge, comprehend concepts. There is some memorization, but the course is not centered around memorization. It is a little bit of a deeper level of learning. You're going to have some research and communication skill development, and obviously it is an online environment which includes some level of self-directed learning, which can be kind of challenging, but you will have guidance from myself and also from numerous resources available to you online. The textbook for this course is an online free OpenStax book. You can find it at the link listed here. It is also going to be in PDF form on the Moodle site. I absolutely do recommend that you download the book, because if you have to go to the link every time you want to read the book, it can be time consuming. This is the cover page. I'm just going to page down to the table of contents. And all of these chapter titles you see there in blue, those are actually hyperlinks. So you can click on a chapter title and it will jump right to that chapter. So that saves you a lot of scrolling. The course materials on Moodle, there are numerous materials on Moodle for you. There's the news forum, which I do use rather often. The syllabus with the course calendar, that you're going to want to make sure that you download and save in a prominent place. Read through the syllabus, put the course calendar in a prominent location, because it's going to have every topic we cover, when we cover it, what days assignments are due, and all of that stuff. The guidelines and the grading rubric for your writing assignments are on the course materials module in Moodle. All of your writing resources, including APA writing guidelines, how to get in touch with the Writing Center on campus. The slides that are corresponding to these videos are going to be in Moodle, and of course your quizzes, assignments. So if you are not already familiar with the Moodle site for this course, you're going to want to get familiar with it quickly, and make sure that your student email address is functioning so that you will be able to receive emails and updates from me periodically. Things to remember: I want to make sure that you know where the website is. My YouTube channel link is on the course materials page in Moodle. It's simply youtube.com/c/Professor Sims. So that's where all of these videos will be. It is also where many other supplemental videos, like study tips and things like that. And the videos, even the lecture videos, are designed to be supplemental. So you have to make sure that you are doing the reading assignment. There are things I cover in the videos that are not in the reading assignments, and there are things in the reading assignments that are not in the videos. So you've got to make sure that you're getting both. Okay. Each Moodle module has the quiz that's due for each lesson, and I will not accept late bonus assignments or late quizzes. Your written assignments, your semester project report, I will take those late, but they are subject to a 25% per day penalty, and that kicks in after the first day. Your exam and project information have their own modules on Moodle, so make sure you have a look through those. Each lesson has specific learning objectives, just like this one. And all of your policies for grading, late assignments, missed exams, again, all of that's going to be on the syllabus. If in doubt, if you're not sure about anything, look first on the syllabus, then look on Moodle. And if you still have questions, you just can't find something, please contact me via email. And my email is located on the syllabus and there's direct links to contact me via Moodle, including the news forum. Okay. Now, if you need to come and visit me during office hours, I welcome student visits during office hours. I have office hours that are designated for 11 a.m. to 1 p.m. every Monday and Wednesday during the semester. And if you cannot come during those times, then you can shoot me an email and we can make an appointment for a time that is better for you. Some tips for studying smarter, not harder. There is a lot of material in this course, and I just want to make sure that you guys understand that rereading, memorizing, rewriting, looking at old stuff, these are methods that were taught to you in K through 12 education, but they're not always the best. The problem is that they are all very time consuming. And if you're taking this class in the lab, and maybe you're taking AMP in that lab, and 2 other classes, and you're just not going to be able to reread everything. You're not going to be able to memorize everything. It's going to be much, much more effective and less time consuming for you to take the materials and transform them in a way that helps you to better comprehend them. So this means like rewriting things in your own words. Take the things from the lectures and the book and try to put them into a format that you would use if you were going to teach it to someone else. Also, self-testing is invaluable. So this literally involves making practice quizzes. Make practice quizzes and use questions that are difficult to you. So this makes sure that you are studying things that you necessarily need to review. There's going to be things that come easily to you, and there are going to be things that are much more difficult for you to comprehend. When you are studying for an exam, focus on those things that are more difficult, and don't waste your time on the things that are not so difficult. And it is very helpful if you can relate things that you are learning now to things that you have already learned in the past. And this can be other courses that you have taken before or things that you're taking concurrently. It's a world-building exercise. Everything in biology is related to everything in other sciences, especially for those of you that are on a nursing or a med school track or health professions track. The things that you learn in micro are going to come back again and again. So if you can relate these things to things that you're learning in your other classes, it's going to be much better for your overall comprehension. And the interleaving aspect has to do with varying your studying. So this is switching between topics or using different resources to study, those kinds of things. It is not a good idea to spend a 3-4 hour long block reading the textbook, because about every 20-30 minutes or so, if you're not taking a break or you're not switching lanes and looking at things in a different way, your brain's just kind of going into sleep mode and you're not going to recall that information. So taking breaks, making sure you get enough sleep, and changing up the way that you are studying the material and the different resources you use. So maybe you're doing a self-quiz and then you look at the video and then you do some reading and then you look at some other topic altogether, and you're taking breaks and you're letting your brain rest in between those activities. It's going to be much, much more effective for your studying. Section 15.1 is characteristics of infectious disease. So this is where they're talking about the difference between signs and symptoms of a disease. Signs of a disease, they're objective, they're measurable. So these are like vital signs, right? Fever, whether or not you have inflammation or an increased white blood cell count, or those kinds of things. Symptoms are more subjective. They're more difficult to measure, but they're important. You know, you do need to make sure that if a patient is coming to you with symptoms, that you use those symptoms in conjunction with the signs that you can measure to try to figure out what diseases they have. But symptoms are more of, you know, things that are difficult to measure, like I'm very tired, I'm fatigued. Pain is classically a difficult symptom to measure because different people feel pain differently. Diseases can be either non-infectious or infectious. So non-infectious diseases are things that are brought upon due to genetics or the environment. So figure 15.2 here gives an example of sickle cell disease. This is a non-infectious genetic disorder that results in abnormally shaped red blood cells. And this is a problem because blood cells can stick together and they obstruct blood flow throughout the circulatory system, including in the lungs. And it's a genetic mutation, so it's not something that is infectious. Now, malaria is an infectious disease, and it also affects the blood cells, but it is zoonotic, and it is caused by Plasmodium falciparum, and it's transmitted via a vector, so it's transmitted from mosquitoes to humans. Communicable diseases are ones that are transmissible between individuals. So I can give it to you and you can give it to, they're transmissible between infected individuals. Contagious diseases are easily transmissible. So these two are very similar. Communicable it means that it's possible, right, to be transmitted. Contagious means that not only is it possible, it's very easy to do. Non-communicable diseases can be contracted via contact with environmental reservoirs or animals. So in other words, they need a vector, or they need some kind of like a fomite, they need some other method to be transmitted, not just person to person. Nosocomial diseases are contracted in hospitals. So these are hospital-acquired infections. Iatrogenic diseases are nosocomial, but they're more specific. They're a direct result of a medical procedure. So this is if you had a catheter and you end up with a yeast infection, something along those lines. If you have surgery and then you end up with a staph infection. Acute diseases are short in duration. Chronic diseases last for months or years or even a lifetime. And latent diseases are those that can lay dormant for years without any symptoms, without any signs, and then they come back. They reactivate. So they generally are not reproducing or showing any kinds of problems for the host, but then at any time they can come back and cause problems later. Characteristics of infectious disease, right? So this has to do with the infection period, and it's very closely aligned to the growth phase of microorganisms. This figure 15.3 shows the progression of infectious disease, and it divides it into 5 periods, which relates to the number of pathogen particles and the severity of signs and symptoms. So the number of pathogen particles, this is directly related to the growth and the reproduction of the microorganism. And then the severity of signs and symptoms, generally your signs are going to be low when you have a lower number of bacteria or virus, and your symptoms are going to be much higher as you have more and more and more, and your immune system is reacting. A lot of the symptoms that you have actually are coming from your own immune system fighting off the microbe. As your immune system starts to win the war, the number of pathogen particles is decreasing, and so is your symptoms. But they have named these periods. So we've got the incubation period, prodromal period, the period of illness, and then it peaks and you start to have the period of decline and period of convalescence. We did talk already about Koch's postulates in the intro lesson, but these are used to determine whether a microorganism is a pathogen, is it infectious, and then what genes contribute to, there's obviously a typo here, but what genes are contributing to the cause of disease. So again, the first thing is you have to be able to observe the causative agent is absent from healthy individuals, but present in diseased individuals. And then you have to isolate that causative agent, and then you can put that causative agent into a healthy individual and see that the causative agent is present in the infected individual and that they have undergone illness as a result. So this is how you confirm a pathogen is the cause of a particular disease using these postulates. Now, virulence, this is the degree to which a pathogen can cause disease. And you can quantify it mathematically. You can measure it by calculating what is called the LD50. So on figure 15.5, this shows percent mortality in an experimental group. So how many people are dying? versus the number of pathogenic agents, how many cells, microbial cells or virins, how much virus is present. This is plotting the LD50, which is, in this case, let's see, LD50, 10 to the 4th pathogenic particles. So this is the point where 50% of the infected individuals are dying. Stages of pathogenesis in order are exposure, adhesion, invasion, infection, and transmission. So this is when an individual is exposed to the pathogen. Then after exposure, the pathogen has to adhere. So it has to attach or somehow become embedded in order to then invade. And then between like invasion and infection, this is where the microorganism actually starts to grow and reproduce. And then, of course, after it has done its job in one host, it wants to go to another host. So transmission is the last step of pathogenesis. And then the process starts again. Primary pathogens versus opportunistic pathogens. Primary pathogens are occurring in otherwise healthy individuals. So it's causing pathological changes in a healthy individual, whereas opportunistic pathogens only cause disease when an individual is already compromised. And this could be in the break in the skin, like breaking your protective barrier. Your skin is your largest organ and it protects you from lots and lots of outside pathogens. Or this can happen in someone who has immunosuppression, so someone who already has a low white blood cell count. Any number of things can cause immunosuppression. Somebody that is dealing with leukemia, for example, or is infected with HIV, and then they're exposed to another pathogen, those would be opportunistic pathogens. Infections can be local, focal, or systemic. And this depends on the extent to which a pathogen spreads. So you have a local infection, like a staph infection. Systemic, though, is going to be something that's in your bloodstream, for example. And secondary infections, again, as opposed to primary and opportunistic infections, these occur after a host's defenses are compromised by a primary infection or an antibiotic treatment. So the secondary infection is quite similar to an opportunistic pathogen, but this opportunistic bit is more of a physical thing, whereas secondary infection seems to be more of like a chemical. Pathogens enter the body through portals of entry, and this shows several different. So your head, your whole head is basically just a portal of entry. Your eyes, your nose, your mouth, your ears. And then, of course, pathogens can pass through the placenta into your anus, vagina, urethra. Of course, any area where you have lacerations in your skin, insect bites, mosquitoes, and passing malaria. And a lot of people already are aware of that, but what they don't think of is the way that pathogens can leave your body. So we're talking about all kind of bodily fluids, tears, milk, vaginal secretions, skin, urine, semen, feces, blood, earwax, all of these ways that you can transmit pathogens. Sections 15.3 and 15.4, these are talking about virulence factors of pathogens. This is when an individual is exposed to the pathogen. Then after exposure, the pathogen has to adhere. So it has to attach or somehow become embedded in order to then invade. And then between like invasion and infection, this is where the microorganism actually starts to grow and reproduce. And then, of course, after it has done its job in one host, it wants to go to another host. So transmission is the last step of pathogenesis. And then the process starts again. Primary pathogens versus opportunistic pathogens. Primary pathogens are occurring in otherwise healthy individuals. So it's causing pathological changes in a healthy individual, whereas opportunistic pathogens only cause disease when an individual is already compromised. And this could be in the break in the skin, like breaking your protective barrier. Your skin is your largest organ and it protects you from lots and lots of outside pathogens. Or this can happen in someone who has immunosuppression, so someone who already has a low white blood cell count. any number of things can cause immunosuppression. Somebody that is dealing with leukemia, for example, or is infected with HIV, and then they're exposed to another pathogen, those would be opportunistic pathogens. Infections can be local, focal, or systemic, and this depends on the extent to which a pathogen spreads. So you can have a local infection, like a staph infection. Systemic, though, is going to be something that's in your bloodstream, for example. And secondary infections, again, as opposed to primary and opportunistic infections, these occur after a host's defenses are compromised by a primary infection or an antibiotic treatment. So the secondary infection is quite similar to an opportunistic pathogen, but this opportunistic bit is more of a physical thing, whereas secondary infection seems to be more of like a chemical. Pathogens enter the body through portals of entry, and this shows several different. So your whole head is basically just a portal of entry. Your eyes, your nose, your mouth, your ears. And then, of course, pathogens can pass through the placenta into your anus, vagina, urethra. Of course, any area where you have lacerations in your skin, insect bites, mosquitoes, and passing malaria. And a lot of people already are aware of that, but what they don't think of is the way that pathogens can leave your body. So we're talking about all kind of bodily fluids: tears, milk, vaginal secretions, skin, urine, semen, feces, blood, earwax, all of these ways that you can transmit pathogens. Sections 15.3 and 15.4, these are talking about virulence factors of pathogens. So when you're discussing virulence factors, you're talking about those stages of parthenogenesis where we're talking about adhesion, invasion, and infection. So virulence factors contribute to the pathogen's ability to cause disease in these ways: to adhere, to invade, to infect. Virulence factors include adhesions, endotoxins, exoenzymes, exotoxins, capsules, antigenic variation, and other things. So we're going to go through some of that. This figure 15.7, this is a glycocalyx slime mold. Glycocalyx is produced by bacteria. In this case, it's forming a biofilm. The biofilm allows the cells to adhere to host tissues. This is actually showing a biofilm on a catheter device. Endotoxin is the lipid A component of a gram-negative cell envelope. So we're going to talk some more later about the difference between gram-negative and gram-positives. For now, just know that an endotoxin is associated with the lipid A of gram-negative cells. So figure 15.13, lipopolysaccharide is composed of lipid A, an O-specific polysaccharide side chain. Okay, lipid A, here's the important bit, is the toxic component that promotes inflammation and fever. And see, both of these are signs, not symptoms. Exotoxins are proteins that are secreted mainly by gram-positive. So endotoxin, gram-negative. Exotoxin, gram-positive. They are secreted by some gram-negative bacteria, but they're usually associated with gram-positive bacteria. This is figure 15.14. Showing a B toxin. B component binds to host cell through its interaction with specific cell surface receptors. The toxin is brought in through endocytosis, and once it's inside of a vacuole, the A component separates from the B component, and the A component gains access to the cytoplasm. But essentially, this is showing the exotoxin that has come from a pathogenic microorganism and how it is attaching to a cell receptor in the host cell. In that way, it is brought into the host cell, where it then reproduces and causes infection. Pathogens can evade host cell immune response. In other words, they can make it to where the immune system can't fight them off. And they can do this by producing capsules to avoid phagocytosis, or by degrading antibodies, or through antigenic variation. So figure 15.17, this is showing capsules that are surrounding bacterial cells. So these are the cells, and then this capsule is made up of glycocalyx, which protects from desiccation, and it protects from being able to be fought off by host cells. This part here, they're showing antibodies. Antibodies normally function by binding to antigens that are on the surface of pathogenic bacteria, and this can induce phagocytosis. Some bacteria make proteases, which break down the host's antibodies, and in that way they avoid phagocytosis. This is an example here of antigenic drift and antigenic shift. This is found in influenza. So figure 15-18, antigenic drift, antigenic shift in influenza virus. So in antigenic drift, mutations in the genes for the surface proteins neuraminidase and or hemagglutinin result in a small antigenic shift change over time. In antigenic shift, there is a simultaneous infection of a cell with two different viruses, resulting in a mixture of the genes. So the resultant virus possesses a mixture of proteins of the original viruses. So essentially what's happening here is the virus is able to disrupt the genetic makeup of the cells such that it no longer produces the antibodies. It can't fight off the pathogen anymore. And in drift, it's a small change. In shift, it's the host cell is being attacked by different viruses at the same time, and you have a large genetic change. Fungi initiate infections through adhesins with receptors on host cells. Some fungi can make toxins and exoenzymes, and they can produce capsules. So fungi can do a lot of things in order to evade the immune system and to improve its ability to adhere and to invade host cells. Protozoa adhere to target cells through complex mechanisms that can cause cellular damage and release cytopathic substances. They can avoid the immune system through antigenic variation and produce capsules. See, so again, you have another example where they have different ways to increase their virulence. Helminthic worms avoid the immune system by coating their exteriors with glycan molecules that make them look like host cells. So they're like little Trojan horses. They're in disguise. And they can simultaneously even suppress the host's immune system. Tricky little buggers. Epidemiology. This is the study of factors influencing occurrence, transmission, frequency, distribution, prevention, and control of disease in human populations. Epidemiology is the science of public health. Morbidity means that you're in a state of illness. Mortality refers specifically to death. Morbidity rates and mortality rates are of interest to epidemiology. Incidence versus prevalence. Incidence is the number of new cases, usually expressed as a proportion versus a specific time period. Prevalence is the total number of people affected and is usually expressed as a proportion, number of infected individuals over a certain time period or a certain area. But the main difference between incidence and prevalence is incidence is new cases, usually during a shorter time period. Prevalence is the total number of cases. Sporadic diseases occur rarely, and generally they're not focused in a geographical area. Endemic diseases occur at a constant, usually low level, and they're prevalent in a population. So this is like the normal baseline level of disease occurrence. But then you have an epidemic. An epidemic is when the prevalence of a disease increases quickly, like as a factor of time. Pandemic is very similar to an epidemic, but the increase is happening over a large geographic area. Epidemic diseases occur suddenly. Pandemic diseases are widespread. An epidemic occurs when a disease outbreak occurs on a significantly larger than expected level. This is showing figure 16.4, a seasonal epidemic set by CDC based data from previous 5 years. When actual mortality rates exceeds this threshold, a disease is considered to be epidemic. So they do measure and track diseases and epidemics and outbreaks and pandemics. So over here you have the percentage of a population versus time. And then there's a certain threshold where if it goes above this threshold, it's no longer considered endemic. It's now considered epidemic. Early pioneers of epidemiology studied disease at population level. Now, this guy here, John Snow, is considered the father of epidemiology. And this figure 16.5 shows a map of an area where he was able to track down the source of cholera. And he found out that it had to do with the contaminated water source. So this is back in the day where they had like a pump, a water pump in the middle of a neighborhood or downtown, and everybody would have to go to the water pump to get their water and then bring it home to use it. He was able to trace back all of these incidences of a cholera epidemic. They seemed to be completely unconnected until he figured out that they all were using the Broad Street water pump, and he was able to trace it back. Florence Nightingale, you've probably heard of Florence Nightingale. She was a nurse in the Crimean War, and this figure over here is showing that there was a number of fatalities in soldiers by month, okay? So that's your percentage of the population versus time. There was various causes for fatalities, but the total number of the people that died in a particular month is equal to the area of the wedge for that month. And the colored sections of the wedge represent different causes of death. So the pink, we have wounds, preventable diseases, preventable infectious diseases is in gray, and the all other is in brown. So again, this is epidemiology. She was tracking the epidemic over time. Joseph Lister is shown here in figure 16.8. He was the first person to use carbolic acid, otherwise known as phenol, during surgery. So he was the first person to understand really the significance of sterilization during surgeries in order to prevent what is now known as an genetic disease. That was Joseph Lister, and you probably have heard of him from Listerine. He was the person that came up with and invented Listerine, which wasn't always used as a mouthwash. I'll let you look that up. Tracking infectious disease: descriptive, retrospective, prospective, analytical, experimental. So descriptive epidemiology uses case analysis, patient histories, happening usually while it's still occurring. Descriptive epidemiology is what they were doing during the recent Ebola outbreak. Retrospective epidemiology is using historical data to identify association with disease and state of present cases. So retrospective epidemiology is generally used when we're looking at reemerging diseases. Prospective epidemiology gathers data and follows cases to find association. It's trying to predict future epidemics. Analytical epidemiology studies compare groups to uncover links between environmental or genetic. So analytical epidemiology is looking primarily at non-communicable diseases. Experimental epidemiology looks at causation in disease or in treatment by manipulating subjects and comparing them with controls. So section 16.3 is talking about modes of disease transmission. Figure 1610 is giving some examples of very common reservoirs of diseases, including human, animal population, soil, water, inanimate objects. So here we've got a very common reservoir for pathogens or doorknobs, door handles. Then of course we've got towels, washcloths, sponges. Your kitchen sponge is disgusting. And then of course things like syringes and other different things that are handled on a regular basis. These are all examples of fomites. Then of course you have contact transmission. Contact transmission it's a classification, so it could be either direct contact or indirect contact. Or with a host, right, that would be direct contact, or with a fomite, so that would be indirect contact. In other words, an infected host has touched an object and then you touch the object, and that would be indirect contact transmission. Vector transmission occurs when a living organism is carrying an infectious agent on its body, that would be termed mechanical, or in its body, that would be biological. And it transmits that infectious agent to a new host. So in this figure, you've got a fly that picks up a pathogen from fecal matter, and it's on its body, so that's mechanical. It's transferred to food, so that would be the fomite, and then the person eats the food. This is a vector transmission. Over here, we have transmission of malaria via mosquito. So the mosquito is the vector, and it is biological vector. Over here the fly is a mechanical vector. Over here the mosquito is the biological vector. Vehicle transmission occurs when a substance such as soil or water or air is carrying the infectious agent. Droplet contact, this is when you're breathing on somebody, you're coughing or you're sneezing, and it transmits your cold virus, for example. Airborne transmission is different from droplet contact in that in order for something to spread by airborne transmission, it has to be further than a meter away. In other words, the bacteria or the virus has to survive in the air, and it has to be able to travel a meter or more in order for it to be considered airborne transmission. So these are aerosols that you breathe in. Fecal oral transmission is really disgusting. This is how cholera is transmitted, for example. You get waterborne pathogens that have come from feces. This is generally, the water supply has been contaminated by sewage, lack of latrines or something like that, and then that water is ingested. And then, of course, you have healthcare-associated infections, also called nosocomial infections, also called hospital-acquired infections. And these are facilitated by medical interventions with a high concentrated of susceptible immunocompromised individuals. In other words, you get a whole bunch of sick people together, and they start spreading around the sickness. In the U.S., the global public health rate, this is 16.4. In the U.S., Centers for Disease Control, also known as CDC, these guys monitor notifiable diseases. In other words, diseases that they can report on. They publish the weekly reports in what is known as the Morbidity and Mortality Weekly Report. The World Health Organization, WHO, this is an agency of the U.S. that collects and analyzes data on disease occurrence, coordinates public health programs in response to international health emergencies. The WHO and the CDC monitor and prepare for emerging diseases. Emerging diseases are ones that are new to human populations or have been increasing recently. So they are upcoming epidemics of a disease. This is figure 1616. And it's saying before the Ebola epidemic of 2014-15, Ebola was considered an emerging disease because of several smaller outbreaks between the mid-90s. And so when they're talking about outbreaks, they are talking about epidemics, but they're small numbers of cases. And then phew, all of a sudden we have an epidemic, and you could also classify this as a pandemic because it's happening all over the world, or it was in 2015. Re-emerging diseases making a comeback. So they had been knocked down, and now they're coming back. And these are the ones that are really scary, because generally when you have a disease that's being suppressed or under control, that's because of medications, vaccines, antibiotics, things like that. For a disease to be re-emerging, it means either the antibiotics are no longer working, so you can have antibiotic resistance, right? Or the vaccines are becoming unavailable or difficult to produce. That is something. Or, you could have people opting out of vaccinating their kids, for example, which is why we have measles that are re-emerging. In the case of chikungunya, we have mosquitoes that are booming in population, and they're crossbreeding even, and they're invading areas where they have never lived before. So swine flu made a comeback in 2009. Probably a lot of y'all remember that. These are particularly scary. Thank you for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.